This is the 7th video in the Fundamentals of Biology, and today we're talking about cell signaling. So what is cell signaling? It is also called cellular messaging, and it is how cells send, receive, and respond to different signals. And the purpose of these signals is to help cells coordinate responses. So signaling can occur in prokaryotes through something called quorum sensing. And this is when signaling molecules surround bacteria, and this allows them to sense their local population density. And due to this sensing, things such as biofilm are able to be created. And biofilm is the aggregation of bacteria on top of a surface. More complicated is how signaling works in multicellular organisms. There are multiple ways that they can do so. One way is through direct contact, and one kind of direct contact is cell junctions. And this can differ between animals and plants. So they're called gap junctions in animals, whereas in plants they're called plasmodesmata. And this allows for free movement of signaling molecules between the actual cells. Another kind of direct contact is cell-to-cell recognition, which is direct signaling between two different cells. Autocrine is when the cell targets itself. Signaling can also occur through short distances. One example is paracrine signaling, and this is when messengers are secreted a short distance. An example of this could be growth factors that stimulate cell growth and division. Another type of short distance signaling is synaptic signaling, which is found in the nervous system, and it is where neurotransmitters are released due to electric signals. However, signaling can also occur through long distances, and this mostly happens through hormones. And hormones are chemicals that travel throughout the bloodstream to travel to a target cell, and this is also known as endocrine signaling. So this works with our endocrine system. So there are three main steps of cell signaling. The first step is called reception, and this is when a target cell, like a signaling molecule over here, will bind to a receptor protein on the cell surface. So this would be the cell, and a signaling molecule will bind to this protein over here. The next step is called transduction, and this is when a receptor is altered and initiates a signal transduction pathway. And finally, through these relay molecules, you will carry out a cellular response, which is where a signal will cause a specific response inside of a target cell. So first, let's talk about membrane reception, which is where a signaling molecule, which is called a ligand, will bind to a receptor. And ligands are very specific to certain receptors, just like enzymes are. So the first type is called G protein coupled receptors, or the GPCR, and here are the steps that happen. So before any signaling molecules appear, the G protein containing GDP is inactive. However, once a signaling molecule reaches the GPCR, the G protein will bind a phosphate group to GDP and activate it into GTP. And this will make it have a lot of energy, similar to how ATP will have a lot of energy. In the next step, the GTP is able to activate an enzyme, and this is used to carry out a cellular response. And finally, GTP will return to a GDP, and it will be recycled just like ATP is recycled with ADP. Another example of membrane reception is the receptor tyrosine kinase, or the RTK. And what a kinase is something that transfers a phosphate group from ATP to another protein. And the purpose of RTKs is to trigger multiple signals within the cell. So you can see that there are many places for signals to be carried out. Another type of membrane reception is a ligand-gated ion channel. And when receptors change shape, it will allow ions to pass through the channel, and this will happen on top of the membrane. However, there are types of receptors that happen inside the cell or intracellular, and they're usually found in the cytoplasm or the nucleus. And keep in mind that the messengers must be able to pass through the cell membrane, so they must be small or hydrophobic. And things like steroids and hormones are able to pass through the phospholipid bilayer. So one example is the hormone receptor complex. And after a hormone attaches to a hormone receptor complex within the cell, it will travel into the nucleus and act as a transcription factor, which can affect how genes are read. The next step is transduction. And transduction is a relay of signals from the receptor to target molecules within the cell. And it is a multi-step process. And by being a multi-step process, it amplifies the signal. In the signal transduction pathway, a signal is transformed as it reaches the target. So one example of transduction is a protein kinase. And remember that a kinase is something that transfers a phosphate group from ATP. But in this case, it's going to activate another protein kinase through phosphorylation. Then, protein phosphatases will remove the phosphate groups from the kinases through dephosphorylation. And this is basically like a molecular switch. And eventually, a protein will be activated in order to carry a response through phosphorylation cascade. In transduction, second messengers also play a role, which are small non-protein molecules or ions that can spread through diffusion. The first example is cyclic AMP. So using a G protein, as discussed before, an enzyme called adenyl cyclase will be activated. The purpose of adenyl cyclase is to convert ATP into cyclic AMP. And cyclic AMP can activate the protein kinase A, which we discussed in the previous slide. Another second messenger is calcium ions. And since there is a low concentration inside the cell of calcium ions, a small change in the concentration of these ions will cause a large difference. And within the calcium ion second messengers, IP3 and DAG are other second messengers to open this calcium channel. So you can see DAG and IP3 are being used to open the calcium channel. And this IP3 gated calcium channel is found on the ER, or the endoplasmic reticulum, and empties calcium into the cytosol. After transduction, a response will be carried out. And the most common type of response is to regulate the synthesis of enzymes or proteins by either turning them on or off, like a molecular switch. And these responses can be coordinated. So you can see in this diagram, the first one is where a pathway will only lead to one single response. However, a single signal transduction pathway can lead to two different responses. In addition, two different signal transduction pathways can crosstalk, where one product can activate or inhibit another response. And finally, if the receptor is completely different, it can lead to a completely different response. Apoptosis is programmed cell death. So if a cell is infected, it will undergo apoptosis. And what happens in apoptosis is the components of a cell are chopped up and put into vesicles, and cells called scavenger cells will be able to digest them. This will prevent enzymes from leaking into the neighboring cells to prevent further infection. And apoptosis is caused by signals from DNA damage or protein misfolding. Finally, let's talk about feedback. So positive feedback is when a change in a certain direction will cause additional change in the same direction. And one example is pregnancy and the uterine contractions by oxytocin. So in this example, the brain will stimulate the pituitary gland to secrete something called oxytocin, and this will cause uterine contractions. And as the head of the baby pushes against the cervix, the nerve impulses of the cervix will transmit to the brain, and that will cause further contractions of oxytocin. The opposite of this is the negative feedback loop, and this is where a body will attempt to stabilize a system by decreasing something that is too large and vice versa. And this maintenance is called homeostasis. In this example, blood glucose levels are managed by the pancreas, and if the blood glucose levels are too high, the pancreas will release something called insulin, and that will remove glucose and store it as glycogen. However, if it's too low, the pancreas will release something called glucagon, and this will break down glycogen and release glucose. And diabetes is something that happens when there's not enough insulin, so homeostasis is not maintained. And as a result, glucose will not be stored as glycogen and glucose levels will be too high. Thanks for watching this video. This video went over the types of cell signaling, the signaling transduction pathway, and feedback.